Hello and welcome to the University of Edinburgh's Research Insights. Thank you for joining us. My name is Hazel Lambert, I'll be your host, and I'm joined this afternoon by scientists Dr. Elaine Emmerson, surgeon Mr Iain Nixon, and Ellie, Jen, Nick and Jen, who are off camera but making everything work brilliantly in the background. Elaine and Iain, welcome.
Hi there.
Hello.
Hi, thank you for joining us. This event is being recorded, but everyone watching, your cameras and mics are off, so we can't see you and we can't hear you. And if you'd like to share this recording with your friends or watch again later, you can find all of them on our website. So remember, this is your chance to ask questions, so the Q&A box is open. If you type them in there, try to keep them short, and you can also upvote so I can see which are most in demand. Now, Elaine and Iain are going to tell us how they collaborate to tackle cancer research this evening. When a person survives head, neck or throat cancer, the same radiotherapy treatment that saved their life often leaves a devastating side effect. Radiotherapy kills cancer cells, but it also damages the healthy cells that create the saliva we use to eat and speak. So together, Elaine and Iain are working on research projects to improve quality of life for people who survive cancer, and to use regenerative medicine techniques to coax cells into making saliva again. We'll hear from Iain first, and then Elaine will tell us about the work she leads in the lab. Mr Iain Nixon is a head and neck cancer surgeon at NHS Lothian. He won't be giving medical advice this evening, but he will be telling us about how this kind of cancer is usually treated. And he is effectively in the hospital and on call. He just had to dash out a few minutes ago and came back in and ripped his mask off ready to talk to us. So if he has to go away, I'm sure he'll come back quickly. Iain, when you're ready, over to you. Thank you.
Okay, thanks for that. And we'll make start. So, hi there, my name's Iain Nixon. I'm an ear, nose and throat surgeon in Edinburgh, and my interest is in treating patients with head and neck cancers. So that's what we're going to talk about today. You might ask, well, what's a head and neck cancer? Well, we're talking really about cancers of the lining of the mouth. The back of the throat, the tongue, the inside of the mouth. There are other cancers that are technically called head and neck cancers. They affect saliva glands and thyroid glands, and there's a whole lot of different cancers. But primarily today, we'll talk about cancers that affect the lining of the mouth and the throat. Now, head and neck cancer is surprisingly common. Worldwide, it's the 6th most common cancer. It's caused by smoking, or at least exposure to tobacco. And in some parts of the world, people chew tobacco, and carcinogens, things that cause cancer, can be in chewing tobacco and other substances that are chewed. Drinking alcohol has an impact, and increasingly we recognize that viruses play a role. Human papillomavirus that causes warts and indeed can be vaccinated against, and Epstein-Barr virus that causes glandular fever, both play a role in the development of head and neck cancers. So as a result of this increased viral impact, even though smoking rates in the West are reducing, actually the incidence of cancers is increasing, head and neck cancer is increasing, and has done for the last 20 or 30 years. So you might ask, how would a patient know? How does it show? Well, really it depends where it is. And I've got a picture here at the bottom right of somebody's tongue, and you can see an ulcer here on the side of the tongue, and that's a way it can present with an ulcer. In the middle here is a picture of a voice box, of a larynx. And these are the vocal cords. And this patient's voice will have changed, and hoarseness is a common presenting symptom. And at the top, this is a picture of the back of someone's throat. You might recognize this bit of the soft palate hanging down. And this patient has a bigger tonsil on, we're looking at the left, but it's their right tonsil than the other side. And so you might get swelling, hoarseness, or ulcers can all be presenting features. And how do we treat it, or who's involved in treatment? Well, it's quite a complex area, and it involves patients' speaking and swallowing function. And as a result, there's not only the doctors that treat it, oncologists who give radiation and chemotherapy, consultant surgeons, but also people who interpret x-rays, who look at biopsies, and people who look after teeth, as well as what we call allied health professionals, clinical nurse specialists, speech therapists, dietitians, and so on, who all contribute towards the complex care required by patients who have head and neck cancer. In Edinburgh here, we provide a service for South East Scotland. Just about 1 million people come through, are covered by the MDT, and about 250 new cases are treated in Edinburgh per year. We're based at the Western General in the Cancer Centre in Edinburgh, but have links across Fife, Borders and Dumfries, and also work with academic partners in the university here and beyond. Treatment for most patients will form either surgery, radiation, or chemotherapy, but most commonly is there's actually a combination of treatment, either surgery and radiotherapy combined, or radiotherapy and chemotherapy combined. So those are the three main modalities of treatment. So we take an example, here's a tongue cancer. You can see at the top this rough area on the side of somebody's tongue, and these can be cut out and then stitched back together. And that clearly has some impact on that patient's ability to use their tongue. In contrast, radiation and combined chemotherapy and radiation doesn't involve surgery, it involves X-ray treatment. And this is a machine that gives X-rays that you can see on the right. And the oncologist will program that machine using a computer and a scan to deliver different doses of treatment. And even though a cancer can be quite small, this is a CT scan of a patient. You can see a large section of that patient's throat will be affected by the radiation treatment. Now you might say, does it work? Well, it's increasingly effective, but there are ways you can predict how likely a patient is to be cured. One of them is so-called stage. And the earlier we catch people, the better they do. Another feature that predicts outcome is sight. And if there are certain areas of the throat that seem to do worse than others, particularly if it presents in the vocal cord because they get hoarse early, we can often more effectively treat patients because the tumors are smaller. And sadly, there's also a link between outcome and deprivation, with more deprived populations doing worse than less deprived populations. So as Hazel mentioned, there can be side effects, and the side effects depend on the precise treatment that's given. And the more intense treatment, the more intense the side effects. And of course, for those patients who survive, those side effects can be lifelong and life-changing. They can change patients' appearance, and they can change their function. As I mentioned before, their speech and their swallowing can be changed. But in particular, survivors of head and neck cancer who've had radiation often find that dry mouth is a very debilitating side effect of treatment. And we're going to go on to talk in some more detail about that and what we're doing about it as we try to meet the challenges in head and neck cancer of improving survival, particularly for those patients who have advanced disease, but also improving the quality of life after treatment, which I think will be the focus of the talk this evening.
Thank you, Ian. Sorry, had a slight problem with my mouse there. It wouldn't work. Thank you so much. Next, we're going to hear from Dr. Elaine Emmerson. And so we know that people experience lifelong and life-changing effects from cancer treatment. And that's where Elaine's work comes in. Elaine is a research group leader at the Centre for Regenerative Medicine. She leads research into ways to repair the damage to salivary glands. And Elaine, what's really interesting about her work is that she works really closely with people who are living with cancer to best understand their experiences and inform the direction of her research. So Elaine, when you're ready, over to you to start your presentation. Thank you.
Thank you very much. All right. Okay, so thank you very much, Hazel, for that introduction. And over the next sort of 10 minutes or so, I'm just going to talk to you about how we're working to restore what most of us consider as a normal everyday function, and sometimes actually is even a bit of an inconvenience, to head and neck cancer patients. So saliva or spit is an essential component of a healthy mouth, and it's really important for a multitude of activities. So if you run your tongue around your mouth right now, you'll hopefully feel that your mouth is quite well lubricated with saliva. And this lubrication is really important for the protection of the teeth and gums from bacteria, for oral functions such as speaking and eating, and also sleeping properly. And right now, my mouth's feeling a little dry, and that's really just because I'm a little bit nervous, but my saliva will quickly return. However, as Ian mentioned, one of the most common side effects of radiotherapy treatment for head and neck cancer is permanent damage to the salivary glands, so they no longer produce this essential liquid at all. So this results in a condition known as chronic dry mouth or xerostomia, which is highlighted by these pictures here, with a dry, cracked tongue and tooth decay because the protective effects of saliva are missing. Over 500,000 people a year worldwide are diagnosed with head and neck cancer, and the majority of these people will receive radiotherapy as a treatment. Of these, 75% develops xerostomia or chronic dry mouth as a result. And below you can see a couple of quotes from patients to really highlight what an impact this has on their daily quality of life. So we looked at clinical trials across Europe and the US, and as you can see from the upward trend of this graph, in the last 20 years or so, there's been a considerable investment in research for xerostomia or chronic dry mouth. However, there's still no permanent cure, and these patients most often rely on the use of constant use of synthetic saliva, oral rinses or lubricants, or even just carrying a bottle of water with them at all times for the rest of their lives to keep the mouth hydrated. So in addition to the impact that this has on patients, it's also estimated that these medications cost over £1 billion per year in the UK alone. So in an effort to develop a permanent cure for chronic dry mouth, we're looking at ways to regenerate injured salivary glands so they can produce saliva again. But to do this, we really need to understand more about the salivary glands. So we all have three major pairs of salivary glands that are located around the jaw, as you can see in this cartoon here. And some of you might actually be aware that just last year, a new pair of salivary glands were discovered that were hidden almost in the centre of the head, kind of behind the nose. If we take a slice through one of the glands, we can see this branching tree-like structure that's quite similar to other organs in the body that produce important liquids, such as the pancreas and the thyroid gland. And this branching network is made up of many thousands of cells that are shown here in purple, called asana cells, that produce the saliva, connected by a huge network of ducts, shown here in blue, that transport the saliva into the mouth. So while this branching network is really the powerhouse of saliva production, there are many other cells that are important in keeping the salivary gland in good working order, including immune cells, stem cells, nerves, and also cells that may negatively impact the gland, like old or senescent cells. And while I'm the one that's actually here talking to you today about this project, I have a team of researchers who are either undertaking PhDs or have completed a PhD, who are the ones that are really truly working in the lab to better understand how all these different cells work alongside each other or even against the salivary gland, and how they might help or hinder us in our aim to regenerate damaged salivary glands. So how do we study regeneration? Well, first we can look at what happened during development, as you can see here in this time-lapse video of a developing mouse salivary gland, in order to try and understand how an organ grows in the first place and the cells that are involved in this. Secondly, we can look at the organ in the immediate hours and days after injury and try and understand which cells make new cells after the gland has been damaged. And in order to do this, we typically use mice in our research. So mice also have three major pairs of salivary glands, like us, as you can see in this cartoon, and they're located in a very similar place around the jaw. In addition, the fact that scientists have fully sequenced the mouse genome many years ago means that genetically modified mice have become a very useful tool in scientific research. So for example, in our studies, we use mice that have had particular cells of interest labelled with a fluorescent protein so we can see them easily. Mice where a particular cell and all of its offspring are labelled so we can see where a new cell comes from, a technique that we call lineage tracing. And mice where we can deplete or knock out a particular cell or gene of interest to understand its importance. So using these models, we've made a number of important discoveries. Firstly, using the lineage tracing model that I just introduced, we found that there's a stem cell in the salivary gland that's capable of making new saliva-producing asana cells. So here, I just want to draw your attention to green cells. So any cell that's an offspring or progeny of a stem cell, so a cell that's been made by a stem cell, will be green. And you can see we have green cells making up these clusters of acinar cells. However, they don't ever replace these ducts here. So this is really important as it suggests that saliva-producing acinar cells are replaced in a very different way to how ducts are replaced. Secondly, using a depletion model, we found that if we get rid of these stem cells, the salivary gland can't replace the saliva-producing acini. And you can see this via the loss of this red colour in this right-hand image when you compare it to the control on the left with plenty of red. Thirdly, we found that nerves support these stem cells, and if we cut the nerve that supplies the salivary gland, we lose these important stem cells. And you can see this by the loss of green cells here in this image compared to the control where you've got intact nerves over here on the left. So this provides a method by which we might be able to help regenerate the salivary gland. Finally, we've shown that there are several different types of macrophages, an important immune cell that's normally involved in getting rid of bacteria and viruses that enter the body, actually in the mouse salivary gland, and that they're really important for the regeneration process. And again, this provides a method by which we might be able to help with regeneration. So what are we currently working on? Well, firstly, we're testing if we can isolate and grow these important stem cells in a petri dish before eventually transplanting them back into the damaged salivary gland. So here you can see that if we take stem cells from a mouse salivary gland and grow them in a dish, they form these beautifully branched little mini organs. So the next step will be to test if we can take these mini organs and whether they can regenerate new salivary gland once transplanted. Secondly, we're working to see if we can mimic the signals that the nerves produce to talk to stem cells and tell them to grow. Third, we know that radiotherapy causes some cells to become prematurely old, or what we term senescent, and this stops them from working properly. So we're testing if we can use drugs to remove these cells without damaging any healthy cells. And finally, as I mentioned before, we know there are macrophages in the salivary gland, but macrophages can act in both a bad and a good way, some causing excessive inflammation and further damage, and some actually helping to repair. So if we can alter macrophages in the salivary gland to be good, we may be able to push the gland to regenerate. And the reason that we're really excited about these findings about macrophages is that macrophages have been shown to be really important in the regeneration of other organs, including the lungs, the liver, and the intestine. So more recently, about a decade's worth of work at our university has shown that a therapy where cells are taken from the blood and then made into macrophages in the lab before being injected into patients can actually reduce scarring and help regenerate damaged livers. Moreover, this decade's worth of work has led to a spin-out company to develop this as a therapy for liver disease patients. So while a lot of our discoveries have come from studying mouse, my collaboration with Ian is really important to compare what we discover in mice to what's happening in human salivary gland. So we do this through collecting small pieces of salivary gland, which would otherwise be thrown away, from consenting patients who are having neck surgery in Ian's hospital. And from this, we can better understand if cells in human salivary glands behave in the same way as they do in the mouse salivary gland when they experience injury, and whether our discoveries are likely to be translatable and of benefit to people who've had radiotherapy for head and neck cancer. And importantly, using a technique where we use different attributes or markers of the cells, we can actually separate out different macrophage populations in the human salivary gland, which you can see here on this graph by these kind of clusters of dots. So we know that there are macrophages there, not only in the mouse salivary glands, but also in human salivary glands. And this is really exciting. So finally, I've already briefly introduced those that are in my research team, but I just want to take this opportunity to highlight that they're the ones that are really working away in the lab to generate these exciting results. This is the building where we work, the Centre for Regenerative Medicine, and this is situated on the Little France campus, adjacent to the Royal Infirmary of Edinburgh. And finally, that we actually can't do any of this work without funding from all of these research charities and research institutions down at the bottom, and without support from patient support charities like the Throat Cancer Foundation and the Swallows Trust. With that, thank you very much for listening.
Thank you so much, Elaine. I have so many questions. I really love getting into the science of it. But for everyone watching at home, remember this is your chance to ask questions. So please do type them in the box. You can give them a thumbs up if you see one that you'd really like to be asked, and that will help me to choose which ones to put to Iain and Elaine. You can ask about anything you've seen this evening, anything at all. Also, hello to Chris Curtis. Thank you for joining us. He said hello in the Q&A. Chris leads the Swallows Trust, which is a charity that supports people who have head or neck cancer and their caregivers. So thank you for joining us. Iain, I'm going to start with a really simple question for you. Why are you called Mr. and not Doctor?
So people who are a fellow of the Royal College of Surgeons, which you have to be a surgeon in the UK, go by the name of Mr. But it was back hundreds of years ago when it became taxed. If you had income because you were a doctor, you were taxed at a higher rate than the average population. So surgeons dodged that tax by calling themselves Mr. And it's stuck since then. Unfortunately, the tax break doesn't still work, but the titles remained.
And Elaine, you said something in your presentation there that just made me go, huh, what? The new salivary glands behind the nose, you said they were only discovered last year. Yeah. How were they found, and how did we not know they were there?
So it's a really good question. In terms of why we didn't know they were there, I think that was quite a surprise. I think we thought collectively as healthcare professionals and scientists that we knew a lot about the body, and so I think this did come as a surprise. But they were actually discovered during a scan for prostate cancer. So one of the things that's funny about prostate cancer scans is that the salivary glands also light up purely because they express some similar proteins. So you can always see the major salivary glands if you do a full body scan of somebody that's undergoing a prostate cancer scan. But funnily enough, last year, a couple of doctors noticed that they could see these other structures that were lighting up, and they didn't understand what they were. They took a more extensive look and found that this was common across all the patients that they were looking at. So they looked at over 100 scans. They then queried whether it was a male thing, because obviously you would think prostate cancer would only be scans for men, but they actually had one female being scanned in this. She also had these structures. And they then went back to the anatomy textbooks and found that actually there were structures that resembled salivary glands, and people had seen them before, but just not identified them as salivary glands. So this actually is a really kind of important discovery and really quite interesting, and amazing. We've missed it.
Are they there in your mouse model as well?
That's a great question. We don't know. So this is something that I think we have to look at to try and understand whether our mouse models, how closely they now replicate the human scenario. But I'll keep you informed.
We've got loads of questions. This is coming in. This is great. The most popular one right now is, why do these kinds of therapies that cure cancer have an effect on the saliva glands?
Do you want to take that, Iain?
Well, the way radiation works is a kind of X-ray, and it's targeted at the cancer. So on one of my slides, I showed a picture, which was a scan, and the doctor treating it outlines the cancer on the scan, and then X-rays are shone at that cancer. And they go through normal tissue on the way to the cancer, because they go through the skin, through all the tissues and into the cancer. And they damage all the tissues that they hit. They most damage the cancer because that's being targeted most accurately. And the surrounding structures get a much lower dose. But they cause a damaging effect on all the cells they have. They kill the cancer cells, but they also damage all the other cells. So patients will get other things like changes to their skin, changes to muscles inside the throat and things like that. But it's the saliva, the lack of saliva, that gives them long-term problems and long-term impact on their quality of life.
And we've got one follow-up question related to that, asking, is there a way to enhance the function of those salivary glands that are spared by more targeted radiotherapy?
Yeah, well, maybe I could answer that as well. The answer to that is that there is no real way to, there's no way to kind of stimulate them at the moment. The hope is, work like Elaine's will allow us from them to regenerate. But I think a few people have asked this question about proton therapy. Well, proton therapy is a way of delivering radiation in a slightly more accurate way, and it may reduce dry mouth when it becomes mainstream, although dry mouth will continue to be a problem, I think, even with proton therapy. So there isn't very much out there, and that's why this is a very interesting research topic.
And that's what brings it back to this sense of regenerative, I can never say that word properly, regenerative medicine, where we're trying to recreate something that has been damaged or lost. Elaine, back to you. We've got a couple of questions here about the images of the mice that we saw in your research, and about whether there are any ethical issues associated with the stem cell element of the research, and again, what you may have had to overcome to demonstrate that it's valuable to use mice in the research. There's two questions there, one about the ethics around using the mice and one around using stem cells.
Okay, so I'll start with the stem cells. So you're correct. There are ethical considerations with any kind of stem cell research. What's really important about what we're doing is we're actually looking at adult stem cells. So they are stem cells that reside or live within the tissue anyway. They're there in the normal kind of day-to-day life and replace tissue as it wears out, or organs as they wear out. And that's a very different stem cell to an embryonic stem cell, which is what we typically consider as being something that there are some ethical considerations to overcome. So an embryonic stem cell has to come from an embryo, so there are issues surrounding that. What we're really focused on is harnessing the potential of these adult stem cells that sit there in all of our tissues, and whether we can either rescue them before radiotherapy, culture them in an incubator and then put them back in and get them to regenerate the tissue, or whether the ones that are still remaining after the radiotherapy damage, whether we can try and kind of stimulate them to regrow and to regrow tissue. So in terms of what we're doing, the ethical considerations are actually much lower than what one would expect for embryonic stem cell research.
And then the animals, so obviously lots of your work is focused using mice. Tell us a bit about that and how you have to go about getting approvals and that kind of thing.
Certainly. So this, again, is something that there's lots of ethics surrounding it, but it's also very tightly regulated. So for all animal experiments that we do, we have to have a specific reason and a specific scientific question. All people that work with the animals have to have gone through a very rigorous training process and to have been signed off as being competent and understanding. And this is actually something that's a legally binding contract. So it's administered through the Home Office, and it's something that we have to take very seriously every single day that we're doing any kind of animal experiments. And then every scientific project is covered by a project license. So we have to justify what the need is for the animal experimentation, why we need to use animals and why we don't have an alternative. And if there are alternatives in place, we should be using them. And so it's really, really well regulated and really tightly justified about the use of animals in scientific research. And it's really only done in the instances where we cannot move forward with scientific research without that understanding.
Yeah. Thank you. And then also in your presentation, you showed a picture of a little pot with a sample in it. And both of you, tell us a bit about that, how you work in partnership. Iain, do you literally give samples from patients to Elaine, and then what do you do with them?
Yeah, sure. We, that's really the collaboration we have, is that we provide salivary tissue from humans, and that's done again in within an ethical framework. The patients who are having saliva gland tissue removed for other reasons, not always for cancer, not really looking at cancer patients solely, but patients who have salivary gland tissue removed, we give a subsection of that to Elaine's team. And so what you saw there was, well, I don't know, that was probably in formalin, wasn't it, with a yellow top? But that's fairly typical. If you've had your saliva gland removed at some point, it will have gone into a pot like that and then gone off to a pathology department. But the arrangement we have is that a small piece can be taken off and then used for analysis.
And then Elaine, what do you do with it once you get it in the lab? What happens?
So those formalin fixed ones that Ian was mentioning, like that little pot, what we do from that is histology. So we can basically slice through and look at the structure of the gland, look at different cell types. So very much like those images that I showed. And from that, we can compare how the human gland looks under a microscope compared to the mouse gland, for example. But more recently, what we've been doing with Ian is collecting the gland fresh. So actually transporting it back to Edinburgh on ice, and then undertaking a technique that's called flow cytometry, which was that kind of graph with the dots that I showed at the end. And that's a method to be able to see specifically immune cells. And so what we're trying to do there is see whether the macrophages and the immune cells that we see in mice that we know are really important in the salivary glands, whether those exact same macrophages exist in human salivary gland. So we've actually been doing these kind of big experiments where we collect the tissue and then process it straight away. So we have to do the experiment that afternoon. So it's, really exciting.
And in one of the pictures you showed, when there were macrophages, so these are these immune system cells, when they were present, you were getting the stem cell regeneration, weren't you? But when they're not present, you weren't getting stem cell regeneration. What's going on there? Tell us what a macrophage is and what on earth it could be doing.
So we're only just starting to find this out. So we've just started doing some experiments where we can deplete or get rid of macrophages again in our mouse model, and find that basically the glands don't regenerate as well as, or they don't replace cells as well as one would expect. And what we're now trying to show is how that communication between macrophages and other cells in the gland, how that's occurring and why that's having that impact. And it's known in lots of other better studied tissues, again, like things like the liver and the lung, that this communication is really important. So macrophages can actually talk to stem cells and other cells within the tissue, within the organ, and that actually prompts them into regenerating. And without that talk, they kind of can't, they're stuck. So we suspect that it's something along those lines, but we don't actually understand yet.
Can you listen in to their chatter? Can you decipher what they're saying to each other? Is that a terrible question?
No, it's not at all, and not yet. It's something that I think in the future, I think will be really important to know, because that's something that, even if we can't change the macrophages, if we can't actually change that physical cell, we might be able to alter the communication, alter that chatter, and try and help regeneration that way.
We've got some brilliant questions coming in the Q&A box, so keep them coming. The one at the top at the moment is, could you possibly implant a fake salivary gland or use technology to stimulate production of saliva instead? Is that something considered?
Yeah, so both of those are really, really good ideas. So the first, the idea of a kind of fake salivary gland is something that others are looking at, more in a sense of, can you grow something from a cell in the first place? So a little bit like that kind of branching organoid that I showed toward the end, the spinning picture. So people are looking at whether you can take one cell and grow something that resembles a salivary gland and then put that back in, and whether that in itself, whether that's enough to regenerate the salivary gland. In terms of any sort of signal or mechanism to make the salivary gland work better or more, that is something that's a current treatment for patients. So that's really one of the main treatments that are offered to patients, besides the saliva substitutes, and that basically stimulates any remaining salivary tissue to produce more saliva in an attempt to sort of overcome the fact that you've got dry mouth. But the problem with that is that those drugs aren't actually very specific. They don't only work on salivary glands, they actually work on many other glands. So people will excessively sweat, they'll have gastrointestinal problems, even a racing heart. So it's something that's really not a very pleasant drug to take. So most people don't want to take it, for obvious reasons. So we're looking at whether we can take those drugs and actually target them a little bit more specifically to just the salivary gland, rather than it becoming something that's a whole body effect. So the problem is, once it becomes a drug in your bloodstream, it's going to have an effect everywhere in the body. So we're seeing if we can just target it to salivary glands rather than everywhere. And that
I suppose that's why it's so important to get those samples, so you can understand what the signature is essentially in the body for those salivary glands. Iain, I have to ask you, you cut things out. That's as a surgeon, you're removing and you're repairing. Do you anticipate there may ever be a time where you could put something back in, like pop a salivary gland back in? Is that the kind of thing that comes up when you're thinking about research in the future?
I suspect it'd be difficult. The reason is that we're getting a lot of questions which can all probably be answered with a kind of single response, that why are they all damaged? Why can't other ones step up? Well, because we've talked about the 3 pairs, or maybe arguably 4 pairs of saliva glands, but the whole back of the throat is covered by tiny little microscopic saliva glands, and so they're so close to the lining of the throat. And the lining of the throat is where the cancer grows. That's why they're involved in the treatment fields, and that's why really almost all of your saliva glands are affected. Maybe not these ones in the back of the nose in every patient, nonetheless. So you've got hundreds and hundreds of tiny little saliva glands, but 6 larger ones. Now the 6 larger ones drain through tubes, and it's hard to imagine how you could implant something and have a naturally functioning tube that allows the saliva to drain out naturally into the mouth, because the tubes themselves are super specialized and have evolved over millions of years. So having just a plastic tube would be difficult. The other difficulty is getting a natural, you could take a natural tube and gland from somebody else and implant it, but to transplant things, you need to deal with the immune system. And generally, after a patient's had a cancer, nobody likes to suppress the immune system. So although in practice, in theory, it sounds like an attractive idea, I think in practice it's unlikely, certainly in the next 10 or 15, 20 years, to be a practical solution to this problem. People are also saying, well, surely you don't need to irradiate all of the glands. And there've been significant improvements in radiation delivery and techniques. People have mentioned IMRT and protons and things. These have been progressively more advanced radiation techniques, which have allowed oncologists in particular to avoid saliva glands and preserve as much saliva as possible. Yet despite all that, it remains the biggest complaint that our patient group have in terms of side effects. And so one of the potential solutions to this, I would say this as a surgeon, of course, is that if we can avoid radiation treatment and cut things out of people rather than irradiate them, that is a practical way that we may well be able to reduce the amount of dry mouth. But the majority of patients still require radiation treatment even after surgery. And so even that impact is limited.
Brilliant, thank you. Now there's another question here that's really timely for both of you, and it's, how has the pandemic affected your work and your research? What kind of impact has it had?
You want to start?
Elaine, how's your lab been?
So our lab actually closed down in the first lockdown. So from kind of mid-March, we stopped all experiments barring ones that were already running. So we were given special permission to go in and finish experiments that had already been started and that would have been a waste of reagents, or particularly animals, mice, if we'd left them. but beyond that, we weren't allowed to start anything new. Nothing was ongoing, no day-to-day research, and we were physically out of the building until the beginning of July. So that was a huge amount of time where all of us were just working from home. And as you can imagine, that's obviously not an easy thing to do as a bench research scientist. Since then, we've been back in and we're able to do some research with much social distancing in place, reduced occupancy in our buildings to make it safe and a safe place to work. But we are really pleased that we are able to get back to some research, but unfortunately, we're still not anything like 100% of what we were before.
Yeah. And Ian, how about for you?
Yeah, I think I would say that it's probably timely to say that Edinburgh University contributed significantly to the COVID effort, and some research groups became the group that did the testing in the early stages and still play a significant role in the test and trace and so on for COVID. From my perspective, because I deal with cancer, things didn't change significantly. I still carried on treating cancer through the pandemic. But in practice, for clinical research, you need patients, and patients, of course, were told not to travel. So it did have a negative impact on studies that required patients to attend for blood tests and things like that. So the whole NHS and to some degree university kind of changed and flexed to try and support the COVID effort. And it's taking a little while to get back from that, but it is happening and research is up and running. And hopefully in the next few months, we'll get back to a relative normality in that regard.
And we're almost out of time, so I'm going to put one last question to both of you. And we'll start with you, Ian, since you're on screen, is if there's anyone watching who's living with head or neck cancer right now, is there anything that they can do to contribute to research?
Well, it depends where you are. Most centres will have research programmes up and running. And if you want to be involved in them, you can ask in your centres about research opportunities. I guess people will be in and around Edinburgh, some of these people, and they're welcome to get in touch with me if you want to be involved. There's ways you can support it by contributing financially, and there are, research funds and so on that you can contribute to. You can contribute to things like Cancer Research UK, which is having a terrible time because all its shops are shut. So it's not got any money coming in. So you can contribute to that as well, and they will hopefully make good decisions about distribution of funding. And following things like this is a great way of staying abreast.
Yeah. Do you know if there's any clinical trials running? Is there anything like that?
There are. Well, it depends what you mean by that. For dry mouth in Edinburgh, we have recently, I don't know, there might be people on this call who are involved in a clinical trial, which is still ongoing. We've actually delivered the last treatment for it, but that was something that we gave a drug that we randomized people to getting a placebo for that was supposedly protective for the saliva glands before they went into radiation treatment. Unfortunately, it doesn't look like it was effective, but that, some of the listeners may be part of that. There are other clinical trials that randomize patients into surgery rather than radiation, and one of the perceived advantages of that, the so-called PATHOS trial, which is open in Edinburgh, is that may reduce the long-term dry mouth. So yes, there are clinical trials that are available locally, and there's always clinical trials on an international front that patients can sign up to.
Great, thank you. Elaine, for you, I know you work really closely with the Swallows Trust and with other patient groups. Do you want to say anything about that to finish?
Yeah, certainly. So the collaboration and communication we have with both the Swallows and the Throat Cancer Foundation is really fundamental to our research and to understanding not only what patients experience, but also what they would like to see. So in terms of our research, helping to shape our research is a really important part. So this kind of two-way dialogue, we could only do that through collaboration with patient charities and support groups like the Swallows and the Throat Cancer Foundation. And in terms of kind of disseminating what we're doing, they're fantastic for that as well. So they have been instrumental in kind of highlighting what myself and my team are doing and getting that out there, because Typically what we're researching is still something that a lot of people don't know even exists. So unless you know someone who has had head and neck cancer and who's had radiotherapy, you might be completely unaware of the fact that this is even a side effect that people live with. And that makes our job actually quite challenging when we're applying for funding, for example, because we're already facing an uphill battle because this is not seen as something that's worthy necessarily of lots of funding, or something that's a huge problem. And because it's not life-threatening, it's also sometimes overlooked as well. So having this network and this support network that do a really fantastic job of disseminating what we're doing and what we're working towards is really valuable to us.
Great. You could put a link to this video in your next funding application.
Absolutely.
And Iain, I think we almost lost you there, didn't we? Thank you so much. I'm so glad that you didn't get pulled away. Elaine, Iain, thank you both so much. Thank you to everyone who joined us for all your questions. Thank you to the rest of the communications team for running the webinar. These events are our way to share the latest in our research with you while we're in this pandemic, but we really want to know what you want to hear about. We've had some brilliant suggestions so far. So I'm going to put up a slide now to menti.com and leave it there for the next few minutes. If you go to the website and put in the code, then you can tell us what topic you'd like to see covered in our final event in March. I'll be back next Wednesday with Dr. Sinead Rhodes, who's going to tell us all about her work with young people who have autism or ADHD, and explain how not all research happens in the lab. So take care, stay cozy at home, and I hope you can join us again in the future. Bye.
Thanks, guys.
Thank you.